All right, Karen, we'll get it underway if that's all right. So, hello all, and thank you so much for coming along to the June installment of our TAGx series. For today, we have a very exciting presentation from Matt Ritchie on characterization of full-length transcripts from single cells. But before we jump into Matt's talk, I just want to quickly spend 5 minutes highlighting how it's possible to extract full-length transcript information out of our single-cell assays. So let's just take a quick second to appreciate the treasure trove of information that becomes available when sequencing full-length transcripts from single cells. Traditionally, our single-cell products have been configured to work with short-read sequences, and as shown in the figure here to the right, that means you're only sequencing the first 50 or so bases of any transcript. This information enables you to perform gene counting and even some mutation detection, both of which being transformative for the world of single-cell gene expression measurements. However, what you've really done in this scenario is compressed all the information about any given RNA transcript down into just a gene count. But what if you could use long-read sequencer to access the information across the entire length of the mRNA transcript? You would obviously retain the ability to do gene counting, but now you also gain the mutation detection along the whole transcript, along with the ability to do isoform discovery and quantification. But at this point, I'm pretty sure many of you are thinking, hold on a minute, 10x Genomics doesn't actually sell any products that enable full transcript sequencing. Well, that's definitely a true statement. You can actually get full-length transcript information from single cells, but it does involve somewhat hacking of the kit. So let's explore how this is possible. For the purposes of today's talk, I want to focus on just the single-cell gene expression and single-cell immune profiling assays, or as you might know them, the 3' and 5' assays. Both of these kits allow you to analyze how cellular heterogeneity contributes to your biological system, or transcriptional profiling at single-cell resolution across tens of thousands of cells. So the best way to explain how to access full-length transcript information is to take a quick walk through the workflow. And here is a high-level overview of the entire wet lab portion of the workflow on the right. We'll start with the process of making gems, as shown here it's shown here in the picture in the video. And you can see that your single cells are encapsulated within droplets, where each droplet also contains a gel bead covered in barcoded oligos. So once the droplet is formed, the cells begin to lyse and the gel bead begins to dissolve to release the barcoded oligos. Now let's examine the biochemistry that occurs within the context of this droplet environment. The barcoded oligos contain a poly-T sequence, which anneals to the poly-A tail of the mRNA transcript. Then the full length of the mRNA molecule is reverse transcribed, and finally template switching occurs to append a common handle to the end of all transcripts for downstream amplification. So at this stage, you can clearly see that we have barcoded full-length mRNA molecules from single cells. The next step is then to break the droplets and get back to working in bulk format within a tube. Since we only have single copies of our barcoded mRNA at this stage, an amplification of these molecules is necessary. So now we have full-length barcoded cDNA molecules, and then hopefully you can already see at this stage it would be entirely possible to sequence these full-length transcripts from single cells. However, in the final stages of our library preparation, we now lose, or as I like to think of it, compress this full-length information. We have to shear down our full-length molecules to much shorter fragments so they're compatible with short-read sequences. Although then we read each molecule with base pair resolution on the sequencer, at the end of the day, we compress this information down yet again to just assigning the molecule to a gene and giving it a count. But what if sequencing the full length of the transcript was really important to you? Or really, how can you access this information? Here's a very high-level overview of the workflow that would be required. Matt will definitely cover this in more detail, but along the top in blue is the standard workflow. Then down the bottom in red is what you need to access full-length transcripts. Basically, you take an aliquot of the sample at the cDNA stage, and rather than fragment the molecules, you simply add sequencing adapters to them and then load them onto your long-read sequencer. There's some custom informatics that are then required, but again, Matt will sort of walk through this solution. But in summary, I hope that you now appreciate that through a relatively simple hack of the 3' or 5' kits, you can now access full-length transcript information from single cells that now add whole new layers to your biological insights. So finally, I just want to conclude on this slide, which highlights the local team here in Australia. So if you're interested in pushing the boundaries of what's possible with our assay, such as full-length transcript analysis, or you just want to discuss your project in general, then I really encourage you to reach out to us. And with that, I'll now hand it over to Matt.
Thanks, Jeff. I'm just going to try and share the right screen. Hopefully you can see. my slides. Yeah, thanks for a great introduction, Jeff. I'm actually a computational researcher, so I'm not going to give a lot of background on the lab side of things. That's much better covered by someone else like Jeff or our collaborator who devised this hack, Jafar. So yeah, I'm really excited to tell you about our work on FLT-seq and FLAMES. And as a computational researcher, we're really excited about getting new types of data. And I guess we think a lot about how to handle the data matrix that you get out of your genomics experiment. And so over the past few years, we've been really excited about single cell technology and have developed lots of open source software tools for this type of data. So it started a few years ago. Okay, well, here we go. So the matrix has usually got genomic features and samples or cells. So you can think of it as a giant spreadsheet that unfortunately you can't analyze in Excel. So we develop analysis tools, and one that we started with, our first foray into single cell analysis, was how do you actually build that data matrix from the raw sequences that you're getting off the sequencer. So handling the barcodes and the UMAPs to keep track of it and do all the bookkeeping you need to do to get single cell information. And then with the proliferation of lots and lots of different single cell methods, we looked a lot in benchmarking. So developing software to sort of handle lots of different combinations of normalization, clustering, trajectory analysis, and so on and so forth. Thinking about different ways of collecting information that might otherwise be discarded, like intron counts are one thing that we wanted to explore in more detail, and we have software for that. And also a nice point and click package that once you've done your gene expression analysis and you have your results, how you can search through them in more detail. But today I want to talk about a really exciting application which has led to some software called FLAMES to explore single cells and get isoform level information, which up to now has been something people have mostly overlooked, I feel. So the challenge here is to put together two really cool technologies from 10x Genomics with the Chromium controller to get your single cells. And also we're very lucky to have access to a nanopore Promethean sequencer, which does sort of high throughput sequencing of libraries that you can create. So the goal was to get full length cDNA and sequence it on the nanopore. Obviously, the advantages of this, as Jeff mentioned, you can get full gene body coverage and really start to look at splicing changes that might be present in different cell types that are in your sample of interest. The challenges that we faced, though, was compared to short read sequencing platforms, we end up with fewer reads, particularly at the time of doing this. Our 10x run on the Illumina NextSeq would typically yield nearly half a billion short reads. But from our nanopore sequencer, it was about tenfold less. So taking that into account, and also the fact that, data analysis, which is obviously our passion, there were fairly limited tools for doing that. I've highlighted here a few papers that are really seminal in this area. scissor-seq is putting PacBio together, I think, with the 10x Genomics and getting fairly light coverage, few reads per cell. RAGE-seq, the work by researchers at the Garvan, which is, again, to do some long read, but this time it's on the T cell or B cell receptor and using short read for the rest of the transcriptome. And work from Lebregon et al., which is very similar in flavor to what I'm presenting today. So our matrix is typically genes by cells for a single cell analysis. And as Jeff mentioned, okay, if you just go and grab the last reads from the last exon of your gene, you're not going to be able to assign those very confidently to different isoforms that might be present for a given gene in your sample. So what we wanted to do is have a separate matrix that we get in addition to our gene level information that we get from our regular short reads, but also can we get some isoform level information from long reads. So here, if we treat our sample carefully and we get full length cDNA, hopefully we can read through not only the last exon or the first exon, depending on the kit you're using, but all the way to the other end of the gene, and quantify that in our different cells as different rows in this data matrix. So as Jeff has nicely introduced, we hack the 10x protocol. And at the point of having full-length cDNA, to account for the fact that we're having fewer reads per flow cell in our long read, we basically say, well, if we're going to have 10% of the reads, we still want an equivalent number of reads per cell. So let's just take 10% of the cells, randomly sample them. Hopefully they'll still fall into the main cell groups or cell type clusters that are there. And then we can overlay isoform level information on the clustering results we get from our short read data. So all of the cells in the sample get short reads just as they normally would, and then 10 to 20% also are prepared for long read sequencing. And so this protocol is available online, and I want to give a big shout out to Jafar Jabari, who was based at the AGRF in Melbourne at the time we started this work. Who was a really generous collaborator who had the idea of how to hack the protocol so that we could generate this type of data. So what are the samples we're profiling? Well, we went to a few, one, not a very biologically interesting one, but great for benchmarking different technologies, is just mixing some cancer cell lines from 5 different individuals. So that's our mixology series here. And we did this a couple of times and did different versions of the 10x protocol, and also gathered some ATAC-seq, single cell ATAC-seq data, again, using a 10x protocol on these data. We had some mouse muscle stem cells from a collaborator, James Ryle, at the University of Melbourne, and they are stem cells that are either control or that have been undergone an insult so that they're injured and they're responding to that injury, and the transcriptional response to that is of interest to them. And we also had access to a patient sample through our collaborators at WEHI in the blood cells and blood cancer division, who this patient was on a clinical trial for getting treatment for CLL. They were receiving venetoclax and they had relapsed on this treatment. So how does this data look? Well, our goal is to get equivalent number of reads per cell, and you can sort of see here in orangey browny color is the number of reads. So these are violin plots of the distribution of reads per cell that we're getting. So we're getting tens of thousands of reads from each type of technology, not always exactly equal. Sometimes we get more long reads per cell, sometimes we get more short reads per cell, but we're in the same kind of ballpark of having tens of thousands to hundreds of thousands of reads per cell, which was our goal, rather than have only maybe a few hundred reads per cell, as some earlier studies had, which obviously gives you a fairly limited view of the isoform variation that is present in a sample. Because we have a count matrix for the long reads and the short reads, if we quantify that at the gene level, we can actually calculate, well, how well correlated are these data? And you can see here that across these different data sets, the correlation is fairly high. So above 0.8, above 0.9, depending on the data set. So there is good agreement between the short and the long read if you quantify it at that gene level. I guess a final view of data quality here is, for our sampling, do we get all the cell types that are there? So here we can see UMAP plots for our mixture experiment. So you can see there quite clearly the 5 main cell lines that have been mixed here, and they're all represented really well. The gray points are cells that have short read data only, and the red points, or orangey, pinky, whatever color that is, points are ones that have both short and long reads. So you can see here across all 3 data sets, we do sample pretty well. And so we're capturing single cell data across the main cell types that are present in our data set, which you would hope to do if you're just randomly sampling gems for your experiment. So this brings us to the interesting part for us as bioinformaticians, which is, well, what do you do with the data? So Jafar's done the wet lab side of things and prepared libraries, and they've been sequenced, and then we have the raw data. So what are we going to do with that? And unfortunately, there were no tools available at the time we started this study. So it was a bit of a dumpster fire in terms of being able to very easily analyze these data, but we were motivated, obviously, because we thought the data would be quite interesting to explore, to develop our own. So our method was called FLAMES, which stands for full length analysis of mutations and splicing in single cell long read data. And the first step of FLAMES is to take all your long reads, and while you're taking account of the barcodes that you learn from the short read data, which very accurately has a high accuracy, so you can get a very good idea of the barcodes present in your data. So you can look to match those barcodes keep track of them, and then first align to the genome using Minimap2. The next step is to correct the reads for errors and cluster isoforms that are highly similar. So we found other tools that perform this sort of isoform identification step tend to find, many, many, many more isoforms than you might think are there. And we looked at spike-in data sets where there's only, there's synthetic spike-ins, the isoform structure is well known, and some other popular methods tended to find, hundreds of new isoforms in data where there really shouldn't be. So we took a more conservative approach here to cluster isoforms and also only keep isoforms that have a decent number of reads supporting them. So then after that step has been done, we will realign the reads to the isoforms and then perform transcript quantification to get our transcript or isoform count matrix that we then take for further analysis. So while we're doing this to the long read data, in parallel, obviously we've got our short reads. So we do the normal thing of doing our QC, cluster those short reads to identify the main clusters. And then those cluster labels can be overlaid on top of our long read data. And then we can use that long read data, obviously, because we have isoform level counts to look for changes in splicing, potentially look for mutations that might be present in different transcripts and see what we can learn from that. So if anyone's interested in that approach, there's software available from GitHub and also from the Bioconductor project. So what sort of things would you want to do with this type of data? Well, this UMAP here shows the short read data. So this is 100% of our cells in this sample from this patient. You can see the clustering that's gone on there. We've got cluster labels there. We've got T and NK cells. Let me do that. There we go. T and NK cells over here, monocytes over here, and this major cluster here are the B cells or the CLL cells that are cancerous. So what we want to do with our transcript count matrix is then go and look. So for different clusters, say for example, cluster 0 and cluster 3, we want to extract out the transcript counts and then do a little statistical test to look for shifts in abundance that happen between the different clusters. So that is the signal of interest that we're looking for. And if we do a fairly simple test to look for that, we find between 500 and 1,000 genes across these samples that satisfy that criteria, which means there are different isoforms in use between different clusters in the data, which I think is really interesting because obviously this is something that for most data sets, you're not able to look at that sort of information. So to give you an example, first we show the UMAP from the short read data. This is the top gene that comes out of that analysis for the CLL2 sample. It's a housekeeping gene, RPS24, and it looks pretty uninteresting, pretty high expression across all the different clusters, all the different cells in this data set. So based on our FLAMES, we're looking for isoforms, which ones do we find, and what are the counts from those? We can generate a little heat map that shows, okay, there are 4 main isoforms for this gene. You can see the gene structure there. The heat map beside it then shows the expression of those isoforms across the different cells where we're color coding them, whether they're normal T cells, NK cells, monocytes versus the CLL cells there in the light blue. So if we overlay this information on the same UMAP, you can see a very interesting pattern here. For example, this first isoform is very highly expressed in the normal cells, in T cells, in monocytes, and less so in CLL. The second one is pretty consistently expressed across all cells. And then the third one here, again, pretty interesting. It's got higher expression in the B cells and lower expression in the normal cells. And so I could bore you with these plots all day. There's hundreds of them. I think they're really beautiful. And I guess they just show the extra level of detail that you're getting from this protocol, which might be really interesting if you want to study splicing in greater detail in your samples of interest. Here's another example. So another category of genes that comes out quite often that you might expect are cell surface proteins. So genes that start with C and D, there are quite a few of them, and they frequently come out in these analyses. So there's another picture at the gene level, so short read data. Okay, again, CD45, fairly highly expressed, consistently expressed across all the cells in this sample. If you then look at isoform level information, you can see that there's 4 main isoforms expressed, and the first of which seems to be more highly expressed in the B cells. But again, there's a subcluster of cells here that has higher levels of expression. And interestingly, we also ran a site-seq panel with this sample. So we have some cell surface whatever site-seq does, I don't know, but capturing different isoforms of this gene, and the antibody, or the ADT, whatever that stands for this gene, is actually specific for, what have we got here? This first isoform. So I think it's targeting one of these internal exons. And you can see that there's also high levels of the CD45RA ADT in some of the same cells that have specific expression of this isoform. So I guess that, you can imagine that if you have certain isoforms of interest, and then there are panels that might target some of those, again, you get extra information that potentially can be corroborated through the site-seq data that you have, depending on whether it's targeting different isoforms of a given gene, which in this case, this one is. I guess another application, so if you happen to have this FLT-seq data and also on the same sample, different cells, but they're taken from the same batch of cells, we did some 10x ATAC-seq. So here you can see an example of a gene. There are 2 main isoforms detected in our data. And you can see if we aggregate the ATAC signal across the different clusters in our data, you can see very distinct peaks. So, quite a big secondary peak in this first cell line, the second one, and the fourth one here, whereas the third and the fifth have low peaks or no peaks. If you look at the expression of these different isoforms, so the one that has this alternate start site and has these extra peaks, you can basically see that these coincide with the cell clusters here, or these are just cells coming from different cell lines that have the highest expression of these genes. So A549, H838, and some of the cells in 1975 have high expression of this, whereas the others have very low expression of that one, compared to the other main isoform, where again, it's fairly uniformly expressed across the different cell types. So I guess that's another example of integrating data from different 10x genomics kits to potentially get a bit of extra support for the isoforms you see, so you can be confident that it's not noise in the data, it's actually a genuine difference in start site for this transcript. So the S in FLAMES stands for splicing analysis. So most of what I've been talking to up to now is about looking at different isoforms expressed across different clusters. The M in FLAMES stands for mutation analysis. So what we're looking for here are mutations that are not germline, so that kind of have shifts in allele frequency across the different clusters. So I guess this is the allele count version of the isoform analysis we were doing before. So from the aligned reads that you have, we're basically scanning through, looking for mutations, and then looking for mutations that are different in the different clusters that we have in our data. So if there's a mutation there, but it's fairly consistent in its allele frequency across all the different clusters, we might think that's a technical artifact or a germline mutation. So less likely to be interesting, versus ones where there's a very distinct shift in allelic expression across different clusters. Well, those are the ones that we think are more likely to be interesting. So if we do an analysis of those, so I guess the first starting point for this is you could say, well, let's take the allele count matrix for the alternate allele for our cell line mixture data, because those are just 5 different donors. It's not really clonal there, it really is germline, but because we've got different donors in there, it will come out reading like how we might expect an interesting mutation would look in a regular sample. So if we make PCA plots from that allele count matrix, you can see that based on these mutations alone, you can separate and see structure between the 5 cell lines that we've mixed for both the repeats of these cell line experiment. If we then go through and look at the patient sample, so this is where it's more likely to be interesting because we do have cancerous cells and we have normal cells, and we'd be very interested to find mutations that might have accumulated in those CLL cells that are not present in the normal healthy cells, and that might be a hallmark of resistance or give us some clue about the clonal structure in this particular tumor. So if we go through and do this analysis, and then we're testing across all the mutations we find to look for, is there a systematic shift in the frequency or in the expression values? Obviously, we're looking at expression here because we're counting the reads that lie on top of these candidate variants we're detecting. So we can find a small number in our data. So here we've got four genes that have mutations in them. One that really stood out and is really interesting to be able to find in this data is this one in BCL2. So this G101V mutation, so called. So this is a mutation that they actually discovered previously using other digital droplet PCR, I think, as well as sort of whole genome approaches. And here what's really cool is we're actually seeing this mutation in different clusters of our cells. So here we go. The red ones have the mutation, the blue ones are wild type, and again, the gray ones are no expression. So you can see here that so again, remember on this side, these are the CLL cells, and there's a systematic shift in the frequency of these cells with this mutation across the different clusters in our data set. So that was really interesting. And incidentally, this mutation reduces the affinity for BCL2 for the drug that the patient is on, and the patient has relapsed. So it's quite cool to be able to detect some mutational signature in our single cell data. So that brings me to the end. I hope that wasn't too short, but in summary, Jafar has hacked the 10x protocol to work with nanopore long reads. We've run a few different experiments using this. There's software to support the analysis of these data, and that was created at a time because there were a lack of tools in the single cell space. Now I think there's maybe 1 or 2 others, but still there's not so many methods out there for dealing with this type of data. So FLAMES might be useful to you if you want to generate this type of data from 10x protocols. And yeah, we've summarized and compared splicing across various different samples and various different cell types, and it seems to have worked pretty well. So yeah, you can find the protocol online, you can find the software online, and you can read more about the paper, which was published last December in Genome Biology, where there are many more results and insights that you can look through at your leisure. So current things we're working on are to really expand this approach to a larger data set. So our collaborators, again, for the CLL clinical trial that I mentioned, have a much larger cohort of patients. So we've been running FLT-seq across many more samples. So that's very interesting. So that data has been submitted and, well, actually accepted, I think, to a nice journal yesterday. So if you want to hear more about that, I think Rachel Thiessen's spoken perhaps in previous 10x meetings, but Rachel's giving a seminar at WeHi in a couple of weeks that you can log into online. So if you want to hear more about applying 10x to study treatment resistance in CLL, that might be a fun seminar to tune into. I guess one of the nice innovations that Rachel's study has added is if you're going after if there are particular transcripts of interest, then she sort of added a protocol with Jafar's help called FLT-seq, which is about targeting genes. So on top of what we've done, which is sort of whole genome, you could potentially have a panel of genes you want to pull out, whether there are mutations in them, whether you think there might be really interesting splicing patterns to find, so that you very much enrich those in your sequencing so you can study those in greater detail. So that's an interesting twist that you can hear more about. We've also run this on other 10x protocols. So I showed you data where we had separately on the same sample, we had ATAC data, and we also had the RNA-seq data from the FLT-seq. But it also works perfectly fine on multi-ohm protocol as well, where now you're going to get both bits of information in the one experiment. And yeah, various, there's a preprint, and I've heard of various researchers also adapting this approach for long reads to spatial, which I think is also really fascinating to get that spatial map of isoform variation. I think would be very cool for a lot of different tissue types. So something to look forward to in the future. And yeah, we have a master's student in the lab, Chongqing, who's currently working hard to continue development of FLAMES so that it works well in sort of larger cohort studies where you have data from multiple samples, such as the data that Rachel will talk about in a few weeks. And yeah, nicer plotting features as well, so that you can easily make some of the figures that I've shown today, which were often compiled in Illustrator, which is annoying, but if you can just run a command and get many of the same pictures, that's way more convenient to drop into your paper. So with that, I'm really grateful to all the members of my lab, past and present, especially Lu Yi, who drove this project as part of his PhD. He's now at the Broad Institute working on spatial biology. So he's very excited about new technology and is learning from experts over there. Quentin, who generated the long read data and has been very supportive of our long read sequencing efforts over many years. So thanks heaps to Quentin and Shani and others who've analysed some of these data for this study. Rachel Thiessen, who was very enthusiastic about applying this technology to CLL samples, and David and Andrew, who've supported that in terms of generating more data, which I think has been really helpful to refine our analysis methods. And collaborators, especially Jafar, who's now at WeHi, but was at AGRF at the time, collaborators at Uni Melbourne in Mike Clark's lab, and James Ryle when he was there, Gordon Lynch. And thanks also to our funders, Chan Zuckerberg Initiative and CSL. And I'll end with some nice pictures of various lab lunches that we like to enjoy as a team, when we have things like papers to celebrate and so on and so forth, students graduating. So with that, happy to take any questions.
Awesome. Thank you so much, Matthew. That was an incredible talk. I'm certainly very excited about long read sequencing technologies being applied to single cell sequencing. Certainly, if anybody's got any questions, feel free to unmute and ask. If not, drop them in the chat. Now, I think we already had one there, which is Matt's nicely addressed, is if you could apply FLAMES to spatial transcriptomics data.
Yeah, in principle. So I guess we don't currently take the sort of X and Y coordinates into account, but that's something we'd be interested to add that capability. So we don't have any data of that nature at the moment. So if anyone has some that we can use to test our software and potentially, again, support, I guess what we like the software to do is support making nice pictures, which is something that 10x, their own software does amazingly well. So I guess we also like to have nice pictures from our data. So thinking of how to make those pictures for spatial data is something we haven't thought of yet, but it's functionality that would be fun to add. So if anyone's happy to share some data. I know there is one preprint out there, again, from, I think, Lebregon, who have similar work in this space for regular 10x protocol, but I haven't seen if the data is available. So, yeah.
And then just from the technical perspective, again, feasibly, there's no reason why it shouldn't be able to apply to the Visium product. It would be for fresh frozen samples only, where we're capturing the actual transcripts themselves. For FFP samples, we use probe-based detection there. So obviously you just be full-length sequencing of probes, which you do on short read sequences anyway. So definitely for fresh frozen tissue samples, that opportunity out there exists. And I'd certainly be excited to see it out in the literature too. I think it'd be really cool.
I think, yeah, I'll put in the chat. I think there is a preprint. So, or if someone else knows it, put it in the chat.
Yeah.
It's from maybe a year or two ago though. So, and I don't think it has come out in print yet. So, yeah.
And if we don't have any other questions, then I just want to thank you so much, Matt, for coming along today for our TAGx event. It was really insightful. We appreciate your time that you'd be able to spend with us and teach us about long-read sequencing on full-length transcripts.
No worries. My pleasure. Thanks for tuning in.
No problem. See you, everybody.